C and D. C and D, so I have to look for 4. Cells are the smallest living structures from which all organisms build their tissues and organs. Okay. This is where it went wrong. Because organisms, you have also unicellular and multicellular. So this is only true for multicellular.
Oh.
Yeah. But in terms of narrowing down, it's a good thing that you narrow down to the 2 lah. Okay. Yeah. That's why I find that the MCQ, this kind of statement, they should make it like obvious rather than a lot of like playing with the language. What could explain the presence of these red colored structures? Phospholipids, right? Okay. So the red stain is actually staining phospholipids. Over time, the cell loses their uniform red color and small red color structures appear. This is pancreatic cells. So what happened to the phospholipid? Okay, electron microscope studies of this red color shows that they are enclosed by a single membrane and contain fragments of membrane. So it becomes like that. So the red dye is around the membrane. So what could explain this? Okay, so it sounds like autophagy. Okay, meaning why? Initially, okay, where are the red dye, right? Okay. So let's say this is the cell, right? Initially, and then here inside you have all the organelles, etc. So they say that initially is uniform red, means that here red, outside red, so everything is like red. After a while, what happens is they found that and then inside shows red. So that means you still have the red outside, but now a lot of reds actually more focused on certain single layer membrane kind of thing. Okay, so inside the cell got membranous organelles which will pick up all these stains. Okay or not? So it tells me that this is autophagy. Do you know what's autophagy?
Not really.
All organelles are being degraded.
Broken down by lysosome.
Okay or not? Yeah. So there are lysosomes that are removing, breaking down damaged cell components. Okay. There are mitochondria with cristae that have been damaged by the products. Okay, they are not damaged by the products. But if the mitochondria are damaged, they will also go through autophagy. Wow. Such a small concept. They actually blow it up.
But why is D wrong?
D is wrong is because this is proteins, right? RER synthesized proteins. The stain binds to lipid. So why should it go to the proteins? They are exocytic vesicles secreting. Okay, A and D, you're saying the same thing.
Wait, but I thought
but
A
at the
would
later stage. So, okay, so you are saying that the vesicles here, right? Because the stain is actually now enclosed inside, not on the single membrane, not on here. So this one is talking about how this thing is enclosed inside. The red stain is inside. So you are saying that here that the red stain inside is actually, you're saying that is actually enzyme that is waiting to secrete outside. But it cannot be enzyme because enzyme do not take up the stain.
Oh no, I mean like the
Protein
the
also don't take up the stains.
Well, I mean like the exocytotic vesicles are made out of like phospholipids. So then that's why like the vesicles will be stained, not the
Yeah,
enzyme.
so it's the outside that will be stained. That means only the single membrane will be stained, right?
Yeah.
Here they didn't say it's a single membrane that's being stained. They said that the contents, the red thing is inside, but the single membrane outside is not stained. Yeah. A small proportion of men, okay, got XYY. In which gamete, which state must this one occur? Okay. So here the XYY means X must come from mother, right?
Mm-hmm.
So the 2Y come from father, right?
Mm-hmm.
Okay. So that means how does the father give 2Y to this boy? Okay, so that's why A and B is out.
Mm-hmm. Oh.
Because it's XYY. So Y can never come from the mother. So that means the person got an X from a normal egg. So the non-disjunction cannot take place in the egg. So that's how I narrow down. So now is how to get 2Y. Okay, so you will see that the chromosome, right, is like that one, ma. Okay, X, Y, right? Got two sister chromatid, right? Joined together, right? Okay. First, under normal circumstance, this one comes here, this one comes here. So this one is Y, this one X. Correct now? And then after that, these two is going to split into Y, Y, and then this one split into XX. This at meiosis 2, this is meiosis 1. Here to here is meiosis 2. When it comes to meiosis 2, this 2 is supposed to split. But it didn't split. So it went on to, one side got 0, the other side got 2, which is YY. Okay, if it's meiosis 1 non-disjunction, how would that take place? If meiosis 1 here means that this one is the X, this one is the Y. Then if they go together means this side kosong, the other side got what? X and Y. So after this one split normally at meiosis 2, they will have XY, XY.
Oh.
And if this one combined with a normal X, it will be an XXY male.
Okay.
Alright, so again, drawing. Drawing helps.
Okay.
You mean you totally didn't draw, huh? You just read the thing and then you come out with your A as your answer, huh?
Yeah.
You're like
no.
super.
I'm just damn confused.
I thought you're really super. But draw means doesn't mean that you have to draw nicely. Just draw a quick one so that your mind can actually see clearly. Okay. Genetics, you have to understand all the rules for genetics. Okay, this one here, 2 genes, different autosome, that means they are like that, telling me that they are not linked. Dominant, recessive, control, color of ear, okay. Dominant B, seal, recessive is chocolate. Dominant D allows seal full. This one is for the full color. The recessive is dilute color to lilac and blue. Wow. Okay, so you have a seal male, seal male. Seal means B. This one is B, full. Lilac is where you must have chocolate first, then the chocolate will dilute it to lilac. So you must have 2D because it's recessive. It must have 2D in order to give the phenotype. Okay, so the lilac mother, done. Seal will be like this. So it's crossed, a seal male having a lilac mother. Oh, this is the mother. Oh, if this is a mother, meaning that the mother would have contributed this and this to the seal male. Agree? It's crossed with a female. So this is a blue female. So a blue female must be seal first, right? So this one must be seal first and then get diluted. To blue. Okay, then also have a lilac mother. This is a lilac mother. No need to redo already. So therefore, the missing allele is actually like that. Agree so far? Are you following so far? Okay. So what is the expected phenotypic ratio between these two? Now I'm crossing this one and I'm crossing this one. Okay, so I will take B, D cross with B, B, D, D. Okay.
Okay.
When you work
I just draw out the whole thing. So I just have to draw out the whole.
Yeah, draw out the Punnett square. What are the possibilities, and then do all the combination.
Okay.
Okay. During the phosphorylation of ADP, what can cause rotation of part of the ATP synthase? Okay.
Yeah, I didn't know what that meant at all.
ATP synthase has two parts, right? The mushroom thingy. Dun, dun, dun. Okay. ATP synthase, this is the proton channel, isn't it? And then this is the ATP synthase. So here, this is the intermembranous space where the H plus is here. So the H plus return to the matrix, right? And the ATP synthase will then do the phosphorylation of ADP and PI here. Alright, so the rotation part is here. This the proton, as it flow through, it's not flow through straight, it flow like a screw like that, all come out like that. So it's actually protons passing through here, ma?
Yeah.
Okay, so this one is out. Okay, This is whether it's chloroplast or mitochondrion. Towards the matrix of a mitochondrion, correct direction. Okay, away from the stroma is wrong. It's from the thylakoid into the stroma. So this part is wrong.
I thought